differential expression statistics. And the fact of the matter is, like, I'm not a statistician. I have taken enough stats to be moderately dangerous. Like, I can make some things up and not be sure if they're right. So I'm going to try to do as much justice to this as I can. If you have more questions, especially if you're on the agricultural campus, Arnold Saxon does consulting, and he has helped us a lot with some statistical questions. So if we have questions about how to analyze some really complex experimental setup, I cannot recommend him enough. He is in the process of retiring in the next year or two. A new person, Liesl Schneider, has accepted the stats professorship in the animal science department, so we will have somebody coming on board who can take his place. I think he consults with people from all over campus. We also have an official statistics consulting group out of the business analytics and statistics department. So business analytics, business analysis, business something in statistics department. And that is run by Bob Minchin. He's the professor, and he has a staff. Sun, I can't remember Sun's last name. She took my class last year. She's also available for free for consulting, which is great. So there's all these statistical resources that you should totally use. So let me kind of tell you a little bit about the evolution of differential expression statistics. I'm going to talk about it in general. I'm going to try to give you some more details about DESeq-2, which is what we're going to be using, and then tell you where the field is moving, because this is a very exciting field with lots of ongoing research. So where we are in the course is we've generated counts. We've used our HT-seq. We've got gene counts for each sample. So maybe we've got a pile of treatment RNA-seqs and a pile of control RNA-seqs, and we've got counts for genes, and now we need to figure out what is statistically meaningful. And at first blush, this looks like a pretty simple problem, right? You're like, oh, this is such clean data, right? We have a count for each gene, each of our samples, beautiful data matrix. Well, like most things in biology and in bioinformatics, it gets complicated quickly. So our first problem is normalizing the data. The biggest and most obvious thing we need to normalize for is that we have different numbers of reads per sequencing run. So when Jean's data was a great example of this, she had libraries with 3 million reads, and she had libraries with 20 or 30 million reads. You can't compare those. You have to scale those back so that you're comparing apples to apples, right? So your counts are not directly comparable before you do normalization. However, there's other types of normalization that can be done. So the first one is based on different lengths of transcripts. If you have a gene that is 10,000 bases versus a gene that is 1,000 bases, even if they're expressed at the same level, you will sample a gene that's 10,000 bases more often, right? So you might consider scaling or normalizing for the length of the gene. Further, you can get a little chaotic with differing ability to map reads to a gene. So we talked a little bit yesterday about you can have 2 genes. If you have a region that is identical, you're going to end up throwing all those reads away because you don't know which gene they came from. So if really only part of your gene is able to have uniquely mapped reads, you would want to scale for that length of the gene instead of the full length of the gene. There's also GC sequencing bias. So if your gene is super out of whack with high GC or low GC content, it's not going to sequence as well as your other genes. So again, you would have to think about how can I normalize, how can I scale my counts to deal with that. However, what the package we're going to use does, and what a good many people are doing right now, is to ignore this. And the way you can ignore this is only compare gene expression changes within the same gene across samples. So you assume that gene A has the same length in sample A as sample B, as sample C, as sample D. If it does have some inability to map reads to some location within that gene, again, consistent across your samples. Some inability to sequence well because of GC bias, that's consistent across samples. So as long as you're saying gene A is moving up or down in expression in my treatments, and do your statistical tests at the gene level, you can safely ignore all of these other things. Some statistical packages will try to help you sort this out. And that would also be significant if you're trying to look at, like, quantify genes in relationship to each other, is when you would need to deal with these things. Okay, so you will still see this used a good bit in the literature, but I would consider it has been at this point, it's becoming less of an appreciated approach by reviewers. So people originally started, they just scaled the numbers and called it a day. But that has some real problems. We mentioned gene link. So RPKM became a very popular way to normalize your counts. And RPKM stands for reads per kilobase of exon per million mapped reads. So you take your count, you multiply it by 1,000,000, and then you divide by the transcript length times the total reads sequenced. So the total number of reads in your experiment. And that gives you RPKM. There are R scripts that will do this for you, but it's also really easy to write an R script that will do this. And this is, you will also sometimes see FPKM, and this is the exact same formula, fragments per kilobase of exon per million mapped reads. And this is just saying we're using paired end reads. So for a pair, you consider it as one DNA fragment with 2 reads. So you're still considering it as a statistical unit. Does that make sense? How many of you guys have heard like FPKM and RPKM? And yeah, these are pretty common. Okay, so why would we not use those? 7 years ago, that's all anybody used. Why have we moved in other directions? Well, these do suffer from some significant problems. So read densities, even after compensating for the total read depth, may not be directly comparable. And this is why. It's because, and we've mentioned this a few times in this class, sometimes you have a really small number of genes that are super, super, super highly expressed. So let's take an extreme example and say one gene is taking up 75% of your sequencing lane. Its expression is cut in half in your treatment, so it's now taking up 37% of your sequencing lane. The other transcripts are going to look really wacky in comparison because they are now getting oversampled because that other gene just left a lot of room for more sequencing to go on. Does that make sense? So a few genes, if they change an expression, if they were super highly expressed, changes the amount of counts that went on for everything else, because they're like eating up less of that lane. So these small genes that change a lot skew the distribution for all of the other genes, especially the genes that are kind of mid to low expression. And this can make a huge, huge difference. So all of a sudden things will start looking significant than art, or things that are significant will no longer look significant. And there's a couple of really good papers, and the DE-seq paper the original DE-seq paper talks about this a lot. So put another way, in case I'm not explaining it well, the proportional representation of each gene is dependent on the expression levels of all other genes if you use RPKM, which is bad, right? Like we actually don't think that that's necessarily biologically true. If one gene is downregulated, that doesn't necessarily mean that other genes are upregulated. So we want to look at our genes as much as we can as independent units. Like the cell could actually be doing more transcription across the board or less transcription across the board, but we don't want to base those decisions on like a single gene. Okay, so our new normalization. Significantly more complicated, of course. We're like, slowly but surely the statisticians are like, no, that doesn't work, we need to add on some new pieces. No, that doesn't work, we need to add on some new pieces. So with each passing year, we get slightly more complicated, but what we think is probably more accurate ways to normalize. So basically we need scaling factors that exclude or reduce the impact of those really highly expressed genes. There's 2 approaches. Well, there's actually a lot more than 2 approaches, but these are the 2 of the most common packages to do this. DE-seq does this basically by assuming that most genes are not differentially expressed. It uses this median of ratios technique to obtain a scaling factor associated with each individual sample. So what this kind of means is that within if you have a whole bunch of genes that are all expressed, like maybe 100 genes are all expressed about the same level, it makes the assumption that most of those are not differentially expressed, and their variability can be applied and used to scale scale their numbers for normalizing. So instead of trying to pay attention to the total number of reads and the whole experiment, it tries to use a subset of genes that are in the same relative level of expression and look at their variability and scale them that way. R uses TMM. People love TMM as well, trimmed means of M values. Basically, they start by excluding genes that have high overall expression or very different expression, and then they compute a scaling factor. Their scaling factor is less flexible than DE-seq scaling factor. So DE-seq scaling factor, it can actually have a different scaling factor for like the low expressed genes, and then the next level of expression will have a different scaling factor in the next. So it's a very flexible scaling factor that gets recomputed at different levels of expression. EdgeR, it's a little simpler. They exclude the genes that have very high expression or very different expression, and then compute a single scaling factor for the rest of the genes. It's worth reading the papers if you're using these. It will help. I usually have to read them like 5 times. But so I don't think either one is bad, but it's just different. So if you wonder, like, I appear to be using the same statistical test. I'm using a generalized linear model. I'm using a wall test. Why am I getting different results from different software packages? It's stuff like this that's causing that. Yes?
So are M values, is that shortening ratio?
The TMM?
So trim means of M values.
Yeah, what are the M values? Yeah, I think it's the trim. How do they calculate the M values? I would have to revisit the paper. I can't remember.
So conceptually, both of these, maybe I'm understanding this wrong. It sounds like what it's doing is what basically does. If you have the things that you model, the stuff you can't model, make a determination about whether what you model is significant relative to the component that you couldn't model, your overall error.
So at this point, we're not computing significance at all. We're not even looking for significance yet. We're just scaling things so that we think we're comparing apples to apples across experiments. So an ANOVA would be looking for significance, like what's involved in, like, are there factors that are gene expression changing due to these factors? Is that significant? This is not a significance. This is not a significance-based test at all. It's basically, it's a normalization. And it's just saying.
But it sounds like we're creating what you would compare to how you would.
Yeah, it's creating a new matrix of counts that you will then do your statistical test on, if that makes sense. Yeah, it's got really complicated scaling factors that are going to change all your numbers. So the numbers you put in are not the numbers you get out at the other end of this. But it's been strongly based in biology and the way that sequencing sampling works. So I think it's pretty good. From there, you need a distribution. Well, potentially, you might want to use a distribution to do your statistics. And traditionally, we do use a distribution. Before 2010, Poisson distribution was often used to model counts. If you take a stats class, Poisson is generally mentioned as like, that's the counting, if you're counting things. Poisson is generally the distribution that you would use. However, it was found that the data was significantly over dispersed. So Poisson has a single variable that predicts both the mean and the variability, but it's not a great fit for RNA-seq count data. Because it's so over dispersed, if you use this, it leads to higher false positives. And now we're mostly using the negative binomial, which is indeed a good substitute for an over dispersed Poisson, and it allows the mean and the variance to be different. So you can have one mean and then you can have a much larger variability. And that this paper was kind of the first paper to come on board and say, yep, negative binomial, much better fit. So the purple is what was predicted, and you can see it doesn't really fit that well. Using the scaling factors and variability in EdgeR, and then using the negative binomial, they got a much better fit. So I put the citation, all the references are at the end of this presentation. However, not everybody has really gotten on board with the negative binomial. You have a lot of options. EdgeR, DE-seq, and DE-seq2 all use a negative binomial. Cufflinks and cuffdiff, which you'll hear a lot about, use beta negative binomial, which I don't know a ton about, but I think it's similar to the negative binomial. There's still a number of packages that use the Poisson. There's a few Bayesian approaches out there. I don't know, I've never tried to use basic. And there's also some people who are saying we really shouldn't be using a distribution, we should be using a non-parametric approach. All of these packages also vary in how they do normalization. I was trying to go through the papers and list all the places, all the different types of normalization I had seen. And they all use different testing strategies. So Fisher's exact, likelihood ratio test, score test, Walt test, posterior probability for the Bayesian, clearly. So you have lots of options, and there's lots of schools of thought on like how you should be analyzing this data. And not surprisingly, with these wildly different statistical approaches, they will give you wildly different results. So it is kind of important to pick something that a reviewer will think is real and that you have some level of trust in. However, I do see publications with lots of different approaches. So just because you choose one doesn't necessarily mean, you just have to hope that like Simon Anders isn't selected to review your paper and you didn't pick his package. That would be bad. But for the most part, you can pick any of these, but you will get different results. So in this one, as you add samples, the number of detected genes can change wildly, but you can see it changes much more quickly for some packages than for others. Number of samples and precision. In this study, they tried to sequence until they thought they got to biological reality. And with a low number of samples, your precision is well below 50%. Moving up as you add samples, all the statistical approaches get better. So this is just to reinforce biological replicates. So the stats will eventually meet at a positive correlation where they all are giving you the same genes. But if you're only using 2 or 3 samples, wildly different results. And false negatives, false positives, it's really hard to tell what's going on with your data. So how to choose? I think I see more publications, especially in the plant world, with DE-seq, DE-seq2, and EDGE-R. I've seen a lot of publications comparing these different techniques that have convinced me that these packages are doing a pretty thorough job of taking into account all of the modern cutting edge thoughts about statistics and giving you the most accurate results they can. With the caveat that right now, almost all of our decisions in RNA-seq statistics are driven by our limited biological replicates. It's just really hard to do statistics when you have a super variable system with a lot of intrinsic noise and not enough samples to actually model that noise and understand it. So I think that's low number of replicates is driving the use of DE-seq, DE-seq2, and EDGE-R. One day, hopefully, we're gonna be able to send off 500 samples, get them all sequenced for like $500, and we're gonna have as many like biological replicates. Hopefully one day will not be our bottleneck. And when that happens, I think the field will be wide open for new statistical methods to take over. The non-parametric people, I mean, I think if you can get up above 10, 15, 20 biological reps, their approach is gonna make a lot of sense. So a little bit more about DE-seq2. Do not try to normalize yourself and then put that data into DE-seq2. Like for example, if you use cufflinks and then you take your expression values and try to shove that into DE-seq2, that would be a very bad idea. Don't do that. So the basic approach is to set up a generalized linear model. So you will need to set up the factors of your experiment, whatever they may be. You can have multiple factors. And the nice thing about a generalized linear model is that it is very flexible. You can have lots of factors. From there, the default test is a walled test, which uses an adjusted log fold change, resulting in a z statistic. And basically, so you can test each coefficient of your generalized linear model. You can also test contrast of coefficients. So if you have like a categorical variable, so like A, B, and C, you can actually do a contrast, like what are the genes that are differentially expressed between A and B, or between B and C, or between A and C, or get the list of genes that just varies according to that factor. So very flexible. The walled test does not need a reduced model, so you just give it one model and test for a particular factor. DE-seq2 also offers the likelihood ratio test. You do need to do a reduced model for that. And this is one of those things, like if I'm on the fence about what kind of test I need to do, I'll often go ask Arnold. But for the most part, if you have a simple setup, the world test is perfectly sufficient. And it will automatically adjust for multiple testing. Benjamini and Hochberg, I think, is the only option in DESeq-2. But it will do that for you. So here is an example of a multi-factor design and setting up our generalized linear model in R. In this case, we can imagine we have 3 treated samples and 4 untreated samples, but we only sequenced some of them in single read and some of them we got paired read. And we want to make sure, since this is not 100% consistent across our samples, that it is included in our statistical model. So factor 1 is condition, factor 2 is the type of sequencing. So when you're setting up, you'll see this, Marianne will show you how DESeq-2 accepts information. But basically you set up a formula. It always starts with a tilde and you put your factor and then you put your other factor. The variable of interest goes at the end of the formula. So the results of this design will by default pull differential genes that look to be differentially expressed due to condition. If you swap to these, you would get genes that are varying based on type instead of condition. So that's how you set up your factors. You can also add an interaction term. Yes, and that's basically testing if the log 2-fold change is attributable to a given condition based, if the log-fold change attributed to one given condition is different based on a second condition. So if there's an interaction between your two factors. For example, if the treatment effect differs based on another grouping variable like species, so maybe your treatment behaves differently in one species than the other species, you can pull out the genes that are causing those differences. And this is how you set it up in R. So type plus condition plus type colon condition, and that would give you the interaction term. It has a giant manual, totally worth reading as well. The paper gives you more background on the statistics and the statistical model, but the vignette is great. It's kind of, they walk you through an example and they show you how to do everything, but super, super, super detailed and very, very, very helpful. And it walks you through how to do all of these things. So one of the most common visualizations you'll see from a differential gene expression experiment is an MA plot. And basically you're looking at the normalized expression versus the log fold change. And so you'll have to read the paper, but there's some shrinkage of the log fold change, which is part of the normalization procedure of DESeq. But this is the typical one that you would see, where you have expression going up. So these genes, each little dot is a gene. You have very, very, very highly expressed genes up here, lots and lots and lots of genes that are very lowly expressed. And then you have the log fold change. So if they're going up or if they're going down. And then the red dots are the ones that are statistically significant. So why are there no red dots down here? It's really hard if you have a count of, like, in my treatment samples, I had a count of 1. In my control samples, I had a count of 3. That's a log fold change. Those numbers are so small, it's really hard to make them be significant. Like, that looks like random variability as much as anything. So it's hard with these really, really lowly expressed genes. It's really hard to get them to be statistically significant. So that's why you see the red out here. When you get more, and then if you have a middling number, a middling amount of expression, you need more log fold change to be confident that they're differentially expressed. And as you get out here and you have very, very, very high expressions with lots of counts, it gets easier to see smaller log fold changes and make them, they are significant. Does that kind of make sense intuitively? And DESeq-2 will give you this plot, which is basically just where it's shrunk the log fold change to say, like, I just don't believe that these really tiny log fold changes are real. But this is the one that people usually expect to see in a publication.
Is the lag one before normalization or both normalization?
They're both normalized. This is before DESeq-2 does its shrinkage. It has this whole shrinkage statistical thing. where it just shrinks the log fold change based on the variability of the data and what it thinks is realistically measured. So it basically says if I've only seen, this is like very, very few counts, like 10 counts, I don't count any of those log fold changes as real. Like if you have 5 and 10, that would be a log fold change, but it's just going to shrink that to 0 because it says those counts are too low for me to believe that that's a real log fold change.
So I just want to know what these plots can tell us how well the data, how many genes overrepresented.
Yeah.
Or it just gives us a roughly idea about how these shrink.
The shrinkage? Yeah, I wouldn't worry about the shrinkage. I just wanted to point it out because if you get this out and you put this in a publication, people are usually going to be like, what? There's no way your really lowly expressed genes did not have a significant log fold change. So you usually want to use this one because it makes more sense prior to a statistical approach. So in this, the only thing, it's not like if I'm just comparing between different experiments. I'm just looking to see, like, in general, what's my distribution of expression levels? And what is my general ratio of significant to non-significant genes? It's a very descriptive plot. It's not giving you, to me, it's just kind of it's the first thing the DESeq gives you where you get to see your data and kind of think about how your data is distributed. So another thing worth mentioning, DESeq operates on raw count data, and then it spits out your statistically significant results, which is problematic if you actually want to do visualizations of your data. So if you want to visualize individual genes, if you want to make heat maps, if you want to look for clustering, there's a number of different output types that DESeq will give you. There's the shifted logarithm, the regularized log transformation, the variance stabilizing transformation. So basically, it's just going to give you these different sets of normalized numbers. Either of these is fine. I can't tell from their paper if they have like a strong preference. They say either one's fine. I think the variance stabilizing transformation is doing a little bit more changing of your raw data than the regularized log transformation. Miriam, do you have any thoughts on these two, or which one you tend to use to spit out numbers?
There are some variabilities on the output, but there is not a strong decision about which one might be better.
Yeah. So from reading the manual, they seem to say either one should be fine. They're just two slightly different approaches. Don't take your raw data and try to make a heat map, is what I'm saying. Because you need to have those normalized and values represented in your heat map. So these are your two options in DESeq2. I think they're both fine. And then you can use those. So basically you pick one and you can say, give me the 30 most highly expressed genes across my samples and generate your pretty heat map for your publication. You can get into like Nature Genetics, right? So, so you definitely want to use your normalized gene counts to generate heat maps. And DESeq2 will do this for you. You can also
Because the other way as well, right? Most like low expressed genes, like if you do not only go upregulated most downregulated.
Oh yeah, you could create a heat map with every single gene. You wouldn't be able to see anything, but right, like it would probably just be a big blue blur. Yeah, you can pick any genes you want, or like your lab's really interested in like the anthocyanin pathway, so you could like just pull up anthocyanin pathway genes and do a heat map of those. So yeah, you can do whatever you want, and you can only do some samples instead of all your samples, or yeah, very flexible. This is why we learned so much R yesterday, so that you can subset your data. You can like pull out the rows you want and shove them into heat maps. All that's gonna come back around today. So another really common thing is sample clustering. I think this is just a really basic hierarchical clustering procedure, but it's kind of nice. You can visualize if your data works out the way you expect. All of your untreated samples cluster together, all of your treated samples cluster together. Maybe there's also some evidence that your paired and single read samples are clustering. So you can plot that and make a nice nice visualization. It will call a basic PCA procedure. We pretty much use this only for quality control. It only returns the 2 largest components controlling variability in your data. But you can do some nice things with like different factors being different colors or different shapes. And again, you would be able to just look at your data and say, okay, it appears that my main principal component is separating my samples based on untreated or treated. My second principal component appears to be separating my samples on paired versus single. Sometimes your data works this way. Sometimes it's too noisy and it doesn't. You can do that within the DESeq-2 package. Okay, so that's essentially what we're going to see in the lab today. I want to give you a little bit of information about transcript level quantification. And actually, I thought about teaching this in this course, and I might teach it next year, because this is, if you have a really well-sequenced reference genome, this is something you're going to want to think about. This is where the field is moving. And that's basically saying, what we're doing in this class is quantifying everything at the gene level. We're not taking into account the fact that genes have isoforms. So this gene might have an alternative splicing variant, and there could be real biological differences. So if you apply a treatment, it could switch transcription from primarily this isoform to primarily that isoform. We'd have a very hard time detecting that with the DESeq method, because we're not looking at the different isoforms. We're testing everything at the gene level. And this kind of everything becomes a little more thoughtful, because you have reads, like these purple reads, these are paired ends, they could be from either isoform. There's no way to tell. These red reads, you feel very confident they have to be from isoform A, because isoform B shouldn't be transcribing this region. Same with the blue reads. So this can be one way that paired end sequencing is really nice, because it can give you enough distant information to identify an isoform correctly. So again, the blue reads, we would say, we think that these 2 paired ends should be no more than 100 bases apart. This exon is way bigger than 100 bases. Therefore, we know for a fact those blue reads originated from a transcript isoform that looks like this. So there are software packages to help you figure this out. But this is kind of a new thing that people really want to dig into and really want to understand about their genes and about their genomes. The software that you would want to look at to do this, Salmon, Sailfish, Callisto, and RSIM are the ones I know of. There are probably others. And you can still use DESeq-2. This Sonesson et al. 2016 paper, they wrote an R package called TX import that will take data, I think the example is from Salmon, and put it into DESeq-2. And this is a really interesting paper, and lots of people believe it. I'm not 100% sure I believe it, but they say if you count at the transcript level and then put the data into DESeq-2 and do your statistics at the gene level, it is more accurate. So gene level results are easier to interpret. They're easier to figure out what's going on. If you incorporate your transcript level estimates, it gives you more accurate gene level results. And one of the primary arguments is that you should be taking into account the length of these isoforms. So by assuming that the gene is the same length in every isoform, that's ruining some of your statistical model. I think this primarily depends on having a well-annotated genome where you actually know all the isoforms of your genes. It turns out that none of my organisms qualify. Like, we may have if we have a genome and gene annotation, we're happy, but really, like, the amount of work that goes into mapping your RNA-seq and figuring out every possible isoform under every possible experimental condition, we just don't have that kind of data. If you work on a model organism, you probably do. If you work on mouse, possibly chicken, human for sure, Drosophila, like any of those models where people have really been digging into the genes for a long time and really know all of the transcript isoforms, I would consider doing this. We had a question yesterday, what if you have a de novo assembled transcriptome? So going the complete other direction where you have like less data. This also presents some statistical problems. Your unigenes or transcript contigs, they can be fragmented. You can have multiple representatives of the same gene. And so fewer reads can be aligned unambiguously, and your counts are being split among lots of tiny fragmented pieces instead of full genes. This can reduce your statistical power, but there are a couple of groups working on solutions to this. The two that I have seen, first is Corset, which was published in 2014, and the second one is RapPlus, which was published in 2016. And basically, you're clustering your individual transcripts into what you think are genes or gene families, and then doing your counts at that level. So if you have a really great reference genome, consider transcript level quantification. If you have a de novo assembled transcriptome, consider using one of these packages. Everything we're demoing in this class is kind of like a straight down the middle, have a reference genome and it's okay. So just want to give you all of your options. I put a whole pile of references. The stats are a lot to chew on in RNA-seq analysis. Some of these are the original program. So this Love et al. Is the DE-seq. I also put the original DE-seq, so you can see how DE-seq changed between DE-seq and DE-seq2. They added a whole bunch of stuff, so they're actually quite different. But also comparisons of different packages are embedded in here. So if you just want to read the papers and verify that, you know, DE-seq it does perform well, it holds up really well in these comparisons of all these different packages. There's about 4 citations in there that'll show that. All right, any questions?
I use DE-seq to about that. So there's an option called P-path. One is parametric local. So what's normally?
I've never used the P-map, the P-H-E-A-T.
No, the P-path. So when you run the DE-seq, you have choice of the P-path. So
Yeah.
Package. So
We just use the default. We don't use,
I think default is a local.
I think, yeah, we do a local fit.
Yeah.
Again, like that's something if I had concerns, I might go ask Arnold. Or I might run it 3 different ways and see if the results change significantly.
Yeah.
Because something like that probably depends strongly on your experimental design and how noisy your data is. Which one is best? That's kind of the story.
Yeah, that's the default local is the most people use.
Most people use them. Yeah. I mean, I think that's what we've
I think also the word test like lab2 is also
we have been mostly happy with the wall test, only if we have a surprisingly complicated experimental design.
One test give you the lab2 folder change of the gene expressions. The other two not so. I think the other option.
Yeah.
That.
So all of these different tools give you completely different testing statistics. All right. Okay, if somebody wants to try to log on to Beacon real quick, just let me know if it's doing it now. Good? Okay, awesome. Yeah, well, there was multiple other mentors. Anyway, maybe they didn't have it set up before. They only thought it was a Monday through Thursday class. Yes. Yeah, you guys have to leave your thoughts with me. Sorry. I tried to fight for you all to get actual research accounts, but they wouldn't do it. So if you want a research account, you can still do it. You just have to register. You go through the whole process of going to class and getting a new job. If you're bringing class over here, let's just do it for real, right? But we're not on board with that. I guess a lot of people take these classes and then they never actually want to use the resource. So they don't want to, that's the problem, they're so expensive. They want you to be committed before you get one.
Okay, so for the next class, we are going to be using the SIG2. For the previous part of the workshop, we have been using some tiny text files. But for here, I have uploaded some data from 6 files, so we will have 3 replicates each. And you can download it here. You can put it on your desktop where you prefer. And I am going to move it to the desktop. And unzip it. So here we have the count files on the same way that we saw them yesterday. On the left, we have the gene names, and on the right, we have the counts. And we have 3 for early flower and 3 for late flower. And there is also this other file that can be downloaded from Phytosome, which contains annotation. We had seen yesterday a GFF file that indicates the genes and their location on the chromosome, but this is different because this will indicate the genes and if they are mapped to similar genes on blood, also trying to get multiple annotation from different databases like Panther or Cake Orthology, Cake Pathways, and also Arabidopsis. And we are going to combine later our results with annotation in this file. So now open R. And the first thing that we're going to do is set our working directory. In this case, we need to point to the unzipped DSEC2 folder. So, for example, in here I can do Command I. With the Command I, you have your path to your desktop, and then you only have to add the DSEC. So this will be the command for me. Okay. So have you all set your working directory? Yeah, so we are next going to load the library. Library. Okay. Well, are you having the same error when you are trying to load the seq? Okay. So then it is easy. You go to packages. What happens then? Larger. Command plus maybe. It doesn't work. Maybe. No, but you have to do ship. Okay.
Oh, 200.
So maybe it is. Okay. So we need the package XML. We're going to click here, install, and just find XML and click install. And that should be enough. So if I try this again, okay, so how many of you still couldn't load the seq? You see, now there is no error message. Now it works. So is there still someone who couldn't load the seq too? Well, so sounds good. So, okay. So now we are ready. We have set our working directory on this folder where we have this folder with the count data. So first, we're going to create a table that will be used by DSEC to load these files in the format that DSEC will recognize. So we are going to first get the file names, only with listing whatever is inside the folder, and like you saw, the names are early flower replicate 1, so we're going to use that information to generate some sort of metadata. First, the sample name, and then the phenotype, which will be early or late flower. So it is file name is list.files, and just given the path. The pattern, if you only have these files, it is not necessary. But if you have multiple files with different names, this might help to get only those files that you're interested in. So going back to R, we have no. Okay, so we have here our file names. This is so to get the other information from these files, what we are going to do is to split the file names with the given character, and then get only the first part of this split. We're going to see part by part. So it is supply string split on the point. Okay, so if you want to split something on a point, you need to use it within brackets because points by default will mean anything. It is like a regular expression, it means one character. So if you want to be explicitly the point, you have to put it within brackets. And then we're going to get.
What does the bracket?
Yes, I'll show. So if we only do this part, if we only split this, this is what we get. So we get a list of lists. And come on. And each string is divided on the point, and you get 3 parts on each item of this list with lists inside. So on the first list, you have a list with these 3 items. The first will contain the sample that we want, second counts, and the third txt. So on the next part of the supply, we're applying the double bracket to indicate each item of this list. So we are going to subset only the first one. Do you get it? So our result looks like this. We're only subsetting the first item. And we're going to do the same, but in this case, we want only the very first part, the early flower, and the separator is the dash. So we are going to do the same, but only changing the split. Okay, got this? So now we are ready to create this data frame that our documentation said that we need to create this data frame with these 3 columns with our sample names, our file names, and our metadata, which in this case is the phenotype. And so we are only going to say data frame with these 3 variables that we have created. We will do that right here later. Right now, it is only a data frame. Okay. Well, we don't need to specify factor. Yeah, this one. Okay.
When I ran that, I got an error.
You got an error running this one?
Yeah, it says error in sort dot list. Text must be at the comment for sort dot list. But I ran it right after mine.
This is weird. Was it like this? I don't get any error. No. Oops. Okay. So here is the table that we need. So with this table, there is this nice, very long function that will get or files from HTSeq and directly import them in a table that will be used by DSeq2. And also, we are going to specify this design option that will directly indicate what are the factors for the generalized model. So in only one command, we will get this my DSeq data that we will use directly. Oops. Oh, come on. I think this one is very long. I will just copy and paste. So here we have all the information about this object. It indicates that this is a DSeq object. Our dimensions that we are having around 26,000 genes, and the 6 samples that we have indicated. And the counts contain the raw counts that we have provided, or name for our genes, and also the names of our samples. So there is an option that it is not necessary with DSeq2, that is removing all low counts. In other packages like HR, they encourage that you do manual pre-filtering of some low count genes. In this case, DSeq2 will do that, will filter out counts with the genes with 0 counts. But there might be sometimes that if you have a big amount of samples, it can be generating an extra amount of data, and the seq will be like a much larger file. And it is then better to remove these lines before starting the process. It would be a little bit faster. So we're going to try this. This is also a way of subsetting. We are going to say the new DataSeq, the DSeq data object, will be overwritten by this one, in which the rows are those that where the genes don't contain counts more than 1. And only to check how this data changed, we are going to use dim for dimensions. So here we see we have almost 27,000, and after we filter, Yeah, well, and we have removed our like 5,000 lines. So we are only keeping those which are non-zero. That's good. Oops. Okay. So now we are ready to do the differential expression analysis. With this, it gets very easy because you only have one function, the D6 function. There are multiple steps that are summarized within this function. So it will get your row counts, they will be normalized, they will be fitted to the linear model from which you have given the design for, and then it will output your results. And it is usually the longest step in the differential expression analysis. So let's do this. Okay, so here you see it is indicating the step-by-step all the different modules in the ESIC. Okay, so it is finished. This one was quick. So now we can see our results. How was it? Error? Do you get the same error? No? This is weird. Okay, it didn't happen yesterday. Okay, so I will try this again. So here we are indicating an alpha because when you get your results, you will have, similar to when you had the e-value in BLAST, it was an overall estimate of how good your results were. Here, there is a p-value. Usually, this indicates that a p-value of 0.1, but usually, if you reduce it, you will get fewer genes. You have to see what it is the p-value that you prefer or that will give the results that are more suited for your analysis. And no, I don't understand. Let's hope it works. Okay, so now it worked. Ooh, this is big. So on my rest, here it indicates that you're going to have the logarithm of the fold change, and there it is comparing the column with the phenotype, late flower versus early flower. So it means if you have a fold change, it'll be, for example, a fold change of 2 means that you will have double in late flower than early flower. And here it indicates the test that was performed, and the number of rows. This one I don't filter, so it contains all the rows. You should have less. And here I'm getting NA values that you will probably want to see, just because I didn't filter in this case the 0 counts. So when I have 0 counts, I get NAs. Also, here it indicates what is the p-value cutoff that has been used, and how many genes you had over or under expressed. This with outliers. In this case, because I didn't filter, it said above that I had 26,000. Here I have 23,000 without 0 counts. It is different. So you see that it automatically filters those genes. How many outliers? The outliers genes usually also don't get the p-adjusted value. It is not enough significant. And also the log counts. So sometimes you will see in this column, the p-adjusted is this one that Meg has said that after performing the first test with the well, I will explain first column by column. On the first column, you get the logarithm of the fold change, which is indicating on this comparison how these genes behaved, one compared to the other. The second one is the standard deviation of the log to fold change. And this statistic is a simple measure. It is only dividing these 2 values. And then we're getting the p-value for the single word test. And then the p-adjusted, in this case, is the Benjamini-Hochberg test. So usually the p-adjusted is less than the p-value, and it's more often the picked one to discard or to get significant genes.
What's the one you report?
Yeah, usually p-adjusted. So usually the ones that are reported just choose a significant value, maybe 0.05, 0.01, and only get those genes that are below this threshold with the p-adjusted. There is one we are going to do a little bit later. So I already said this. Here, this is only for checking that if you get your p-values and you apply the method Benjaminian software on your own, the results will be exactly identical for those that are reported by the SIG and those that you are getting on your own. Because you can choose any other test of your preference. It is only the default one. So, okay, yeah. Here. Okay, do you want to test it? Yeah. Oops. Yeah. Okay, so analytics 20 to be better seen. Oh, this is weird. Okay, no, this is different. Yeah, so you see that there is a difference from the p-values to the p-adjusted. P-values that were significant, more significant before, usually get less significance when you adjust them. Well, it should be different on your screens because I still get NA values. But anyway, the numbers are the same. When you have a data frame, it is very useful to use the dollar sign so you can select easily one of the columns. Okay, so we are going to move into the next section. So first, visualize our counts and our significant genes with an MA plot. For this, we only have to use the results that we're getting. I have indicated the title for this plot, and I have adjusted the limits to show all the points within this plot. Although if I did only a plot MA my res, it should work exactly quite the same. Plot MA. Oops. It should be adjusted. Okay, so we are having 4,000 genes, and they're spread around both sides of the log fold change. And like Meg said, they're mostly in the middle and upper counts. For lower expressed genes, this fold change with the 3 replicates that we have given are not detected. Probably increasing the number of replicates would start also spreading the significant genes below this threshold that we are seeing now. And usually you will want to use your results in other platforms, or maybe to send them to your collaborators for publishing, or using them in Excel for other things. So we are going to see how to export our results for only our significantly differentially expressed genes in a table. And we're also going to merge the annotation that we had seen before. So our results are going to have much more information now. So first, bless you, we're going to subset our results based on the p-adjusted value with this 0.05 cutoff. And run. You see in here, we have 4,000 differentially expressed genes. So, for example, let's see. What would happen if we decrease this value to 0.01? We would 2,000 genes, almost half. So that's what I was saying before about playing with the significance. So you can try to adjust a little bit how many differentially expressed genes you want on your results. And then we're going to load the annotation data that is on the same folder that we had set our working directory. And for that, we are going to use the read CSV function, which is a wrapper of the read.table data. And we have to give the name of the file. We are going to indicate that it has a header, that it is separated by tab instead of by comma. And also we are going to indicate not to convert the strings as factors. Well, this is like a long name. I don't want to do it wrong. Oh, fine. Okay. It will be messy. But so this is my annotation file, how it was loaded. It contains some empty columns. So this is something very important when you have a file that doesn't contain data in all columns. This wrapper can parse well the file, so they won't shrink these columns and move the others where they shouldn't. So they maintain the Go terms are here, same for the Arabidopsis definitions. So we have to check that it looks like we want. And it does. So we see, well, this locus name contains a name that is very similar to the name in my res. So we are going to do so you see the row names on my results are like this. So we need to have this information before we can merge both files. Oh yeah. With the annotation file, yeah, I only indicated to here. Yeah, you see they are the same locus name because they are different transcripts. So we need to merge this and only get one transcript for each locus. Otherwise, it will try to pull all of them, and we only want one in our results. So we are going to use the function duplicate to remove these columns. With this exclamation, we are going to indicate not to print the duplicated lines, so only to keep the unique ones. And this comma 2 indicates on the second column, which is for our locus names. Yeah, and I don't want to. Yeah. If you had done then, you could have checked the dimensions of the annotation file before and after the filtering. I didn't. So to merge these two tables, we are using the function merge, where we indicate the first table. These results significant is a DSEQ data object, so we need to convert it into a data frame, and you only indicating as data frame, that's it. And then the second find is annotation. On what column in the first file are the row names, or the column in the second file, like we saw, is the locus name. We're indicating that we want all the rows from our significant result after the seq, and that we don't want all the rows on the annotation file. And that prepares the file, and then we can write the table. And yeah, so we write our table, we indicate our output file, for me, I prefer the files to be comma tab separated. Sometimes you can specify if it is of your preference, comma separated. But when you try to load these files into Excel, I don't know why by default, when it tries to load CSV files, it searches for the semicolon. So it may cause some problems. So that's why I always prefer tab separated. And also we are selecting not to print the row names, because if you see the first part of these results, the row names are only numbers. And now the gene names are in this column that is now called row.names. So now going to the Finder, this is very tiny. Okay, so this is our text file that we have generated, where the names, as you see, are quoted and the numbers aren't. There is an option for you to remove these quotes. It is on indicating quote and quote. It means there is nothing in between. And yeah, this is of your preference, or maybe depending on what you want to use this downstream for. So here you have all the information together. Here, there are some points if you want, because one might have many biological questions, or you may be interested on what are the overexpressed genes or underexpressed genes. You may want to subset your file to capture only those. So here are some options that you can test on your own. And then see how many genes do I have on each list. Like I was saying before, if I change the alpha to 0.1, we saw before that our genes were reduced to almost a half. And also, what is the range of p-values or average of the fold change? So there are many things that you can be interested about your results. Or maybe if you're interested on a certain pathway, how to subset your results based on a given set of gene names. Our next part, we're going to see some visualization options of our results. But first, like Meg was saying, we need to transform our data. We have the two options, the regularized logarithm of the VST, variance stabilizing transformation. Both will work well. There is not such a strong preference one after each other. The results look slightly different, but they are both indicating the same things. So for my examples, we are going first to generate both, but I will plot only, well, here on these examples, only the R logarithm is used. So you can try to do the same things, but with the variance stabilized transformation. So first, we are doing the R log our data and fitting on the mean. well, they are both fitting on the mean. Yeah, these steps of transforming the data also may take a while. It depends. If you have larger data sets, it may take long. Also, they both don't take exactly the same time. I don't remember now which one took a little bit longer. But lucky for us, we are working on a smaller data set, so it shouldn't take very long. Okay, so what it runs? We are going to see sample and gene clustering using the heat map. The assay will extract the count from our raw data set object? No. From our transformed data, from transformed data. This is a way, before we have used the subset function, but we can also indicate within brackets, it is first columns, and second, the rows. So we're going to subset the columns based on what columns are in our object, my results with the significant p adjusted. And also they will be ordered based on the p adjusted values. And here, what I say is only get the 50 top significant genes. If you want, you can change this number. So did you understand what this function is doing? Because this is functions within functions. So we are getting our numbers from the logarithm transformed data and only get those row names that are in the 50 most significantly differentially expressed genes. Here, if you remove this, you will plot all 4,000. You can also try. And then here on the second part of the comma, we are indicating select all columns. So we are only subsetting by the rows. Yeah. Have you tried to load the library field map? If you don't have it, you can go to packages, install, and install it from here. So now it is very easy. We only have to use this function with our object that contains the heat map. Cool. Yeah, and the bad part is that all the row names are very stuck together. But here you're seeing how they're changing. Those with lower transform data in early flower or late flower, and how in some points they are different. They are lower in early flower than late flower, or reverse, more abundant. And yeah, so this was only done with 50. Like I say, you can change the number here, and you can select as many genes as you wish. Yeah, also, I forgot, there are clusterings in both sides. So from our data, it is very good because it is clustering or both phenotypes, early flower and late flower. So they indicate that they are similar in between groups. And also this clustering of genes help us to realize which one are more differently expressed. Well, let's continue. So next, we're going to see the sample to sample distances with the heat map. There is a lot of code here. So first, we are going to calculate the distance of the how was it? Transpose matrix. So if right now the distance would be calculated between genes, but we are going to calculate the distance of the transpose matrix, which contains the sample. So we are going to see how far each sample is in relation to their genes. And then we're going to transform these distances into a matrix. And just for better visualization, remove the column names of this matrix. And here I have changed the default colors, and I have generated a custom palette where I have used this function of color from palette and selecting from blue to white with 10 steps. And finally, load it with the fiat map function, supplying this color palette. And this is a lot of code. I will just copy and paste. Here, this one. So I'm selecting colors from the blue to the white in 100. So it's going to be gradually changing from blue to white in 100 steps. Yeah, if you know, we can select a block of code and run all it at once. So this is our result. If I can open it. Yeah. So it is again clustering the sample, but now it is only clustering samples. We don't have the genes here. This is the color scale that I have generated. And in here, the lowest, I think, is the most, this is distance. So the lowest distance indicates they're exactly the same. At highest distance, so less related. So you see that the blue is on the diagonal, indicating that which one is late flower, that the first one would be also the late flower replicate 3, and so on. So here we can see that our late flower samples are much more related than on early flower samples. And mostly our replicate 3 of early flower is more distant to the other two, which are more close because the blue is brighter. And also late flower compared to early flower, these squares indicate that there is a long distance on the gene profiles of each other. If you realize this is a diagonal matrix, so maybe you can try to find a way to only plot the lower or the upper part and use the other section for another kind of plot. That it can be seen in some papers. But here it is symmetric. And our third plotting is principal component. Here we don't need to do anything special. We only indicate to plot our principal components of our data, only that we are going to select those that are in our significant results. And we are indicating for coloring that our internal group is the phenotype. Error. Oops, yeah. Okay, so here, this is another representation where the samples are separated in relation with some principal components that are trying to reduce the complexity of the whole matrix with the counts. And with the first plot, we are getting 84% variance, which is pretty good. It is separating very clearly both groups of early flower and late flower. And our second component, it is only 12% variance. In this case, it is not clear what it is separating because our late flower are together and there is something different in the early flower samples. Sometimes these kind of plots are very consistent with the phenotype data, but other times you will see that the plots are distributed in a way that it is not so clear what factor might be driving these differences. So is there someone who didn't get to this point? Okay, so on the next part, after we have been seeing how to do, so we have seen how to import, load our HT6 data into R, transform it into a DSeq object, how to do DSeq, and then different ways of visualization of results. So sometimes this was an easy example. It will run fast if you run it again. But if you're working with a larger data set, sometimes it might take much longer. So there's a way that you can save your object that you have created in the workspace into a file. So next time you only load this file and you will already have these variables. For example, here, you have the name of the variables in this upper part. So here we have file name, first time, well, what are important, for example. The most important will be probably my results or my result significant. Maybe also it took quite longer to do the R logarithm. So we can select either the whole workspace or only some objects and save them into a file. So for the example in the wiki, I just selected one file, but you can comma separate as many as you want. So you only have to indicate the variable or variables and the file, and that's all. Mm-hmm. Even though this is very tiny. No. So here is the file that I have created. I think that this should be uppercase, but it's okay. For the lower part, at the beginning of the course, I was saying that we wouldn't get to the goal enrichment analysis, but it is very straightforward from the point that we are here. So I think you would like to have this, because many times when you get your results, like you say, if you already have the functional annotation, like in this case, it is very easy to import the result. Or maybe if you had your Trinity assembly, or you had run BLAST or interpret scan and had Go terms, you can parse them and use for visualizing in other tools. And that is something very helpful, mostly to interpret your results. They're also suitable for publications. So we're going to use Agrigo in this case. And Agrigo accepts to have custom references, and they have their own, I'm sorry, I'm searching for here, their own format. Not here, please. On the tools, oh, I cannot amplify. Yeah. Okay. So for customized annotation, they say first indicate your gene, one space, and your Go term for your query and for the reference. So let's do that. Okay. No. Here I have a small explanation of the commands that we are using for this. That again, we are using the string split that we had used before on our Go annotations. Why? So if we do head on our Go terms, we see our format. So we see here that we have our Go annotations, not necessarily one per gene, and also not all of the genes contains annotations. So first, what we are going to do is to split these Go terms on the separator, which is a comma. So we'll get list with as many genes as with as many Go terms as in the for each gene as there are. And then we are to generate a data frame replicating each locus name by the times that it contains Go terms. So if one gene contains 2 Go terms, this gene will be replicated twice. If it doesn't contain any Go term, it won't appear. That is what the supply means. For only calculate the length of the number of Go terms for each gene. And then here on list, remember before that when I did a split, we had a list of lists. So it will put all of the contents into a single list. And with this, we are creating a data frame with only 2 columns. The first column with our replicated loci, and the second column with all the Go terms. And they will match which Go terms is on each loci. And then we're only going to write our results into a table where our separator will be the space. And with only these 3 lines, we're going to get the format for AgriGo. And we are going to do the same for the whole annotation, so we can get our reference, and also for significant genes. So to do it a little bit faster, I will also copy and paste. And the same for my results. Here, what I was indicating, remember before we said that our annotated results, the locus names were in a column that was called row.names. So that is the difference. And run. Okay, so this is the last piece of code in R. So now with our results, which should be here, new to files, go on go, they're very tiny, in our required format. Command plus. We will use this into AgriGo. So okay. So on top, I select customize annotation, and I'm going to only copy and paste all the genes on our results. So I think select all and copy it. And here, paste. And here you can also do the same, copy and paste, or you can upload the file. I think that I'm going to copy and paste. Okay. Oh. If we see on the advanced options, it indicates a statistical method that is using, also a multitude adjustment method. By default, it indicates utility, but you could also indicate Conferroni, Hochberg. We're going to only let the default parameters, and also the minimum number of mapping entries. And we are going to use the complete gene ontology. We're working with plants, so we could also use the plant go slim terms, but they're usually much more general. So we will let the complete go. And we only have to click submit. So, yeah, it is working. So you see that you're giving a job ID. So when the job is finished, oh,
I know we're using the website right now, but are there any R packages or other tools you would suggest to do?
there are a lot of R packages, but I didn't use them a lot. Mostly because I'm working with blueberries and the annotation is not very good. But if I can think of any, I can tell you later. Yeah.
Never used the EC to do it. We've only used other packages like Bingo, State, or you want to look, yeah, the prevalence, and you see if you do get a term enrichment between your samples or from your samples to against like all genes. You have to aggregate and count. Yeah, I don't know. I've never done these two. I'm not saying it's not possible.
So here, yeah, I only saying go enrichment in our package. There is stop go, go plot. They are in here, comp go. I didn't use them. Go express, but you can check. So continuing with this, the interesting thing about AgriGo is that here you will get your own reference number and you can share it. It is useful for 1 week. So if you have other collaborators, you can be sharing these results with them. Also here you get some of the information that you had used on this analysis, and you can get these two kind of graphical results, selecting your format, also how many genes you want, the direction, or mostly if you want biological process, cellular component, or molecular function. And same for Go Flash. The difference here is For example, you can only plot one of the 3 components at a time on the first plot, but you can combine the 3 of them in this plot. Yeah, let's take a look, for example. And the last thing that I'm going to indicate, what did it say? Okay. So here is everything. For example, metabolic process is very high, catalytic activity and binding. Yeah, but they might seem a little bit general and go back. Something that I like is this part, ready go. What this does is it clusters the Go terms by semantic space. So it is like if they are semantically related, they are performing similar functions, they will be aggregated. So it automatically adds the results from AgriGo, so we don't have to do anything else. Maybe if we want to change some of these parameters, but you can also leave them on the default, because it will also select the allowed similarity, and you can, these results will be filtered by p-values. And start ReviGo. And in here also we will have the 3 components in separate graphs. This is the one for biological process. This is the interactive one. And we can check and keep or deselect the terms that we want to be displayed. No, this is a little bit bigger. And after you have seen this, there is also another interesting option in here. You can generate an R script, which will create a plot very similar to this one into R, and a plot that you can save and use for your publications. This R script is very easy to use because it already contains all the information. And well, so find the pretty good. Yeah, so in RStudio, we open so here you have all the information required to generate the plot. You can also, if you learn how to tweak it, you can make some changes to make it look more like you would like to. And if I select everything and just run, if everything is correct, oh, now just waiting for it to resize. It is big for this screen and this, but here you get an upload. And this is all for this part. Do you have questions? Okay, so are you going with the batch script?
Yeah, we don't have a ton of time. I'll try to run through 2 super quick examples of the loop and the batch script. If you guys want to take 2 minutes, then take breaks to take a break today. While I get set up, that's fine. You got get back in here, just log in the weekend. Oh, and send me a PCA plot if you're an enrolled student. Just attach it to an email.
Besides, we used to do our DSafe today is from generate from yesterday after we finished the HTS.
We had pre-computed a bunch of data sets from Geely's larger data files. It was still a subset, but yeah, so we had just run through the pipeline on different sets of files so that you guys would have something you could do differential expression.
I know where the go term from is after we finish the HTS, it will give us
where can we find the go terms?
Yeah.
So you can, if there's a reference genome, you can go find them from a community database. That's where we got the go terms for Peach from like the RoseAC genome database. You can generate your own using InterProScan or Blast2Go, or there's a number of functional packages. We usually try to teach InterProScan to generate go terms, but we haven't got it working on this system yet. We're trying. So if you're interested in using it, eventually we will eventually have it figured out.
I
but
think
there are
after
ways.
the HTS to the step 7, it only give us the gene map to
The counts.
Gene counts, right? There's still a step links the gene to the GO, right?
Yes, you either have to go find that from another group who's done it, like if somebody annotated the genome and they annotated the genes with ontology terms, or you have to generate it yourself. And if you generate it yourself, my recommendation is InterProScan or Blast2Go. I'm trying to think what else. KEG will give you go terms. Yeah, that's how I do it.
I see.
Yeah. All right, so we do not have much time, so I'm going to fly through this. And this is more like if you don't even want to get on Beacon and try this yourself, that is okay. Because at this point, I can't teach you to code in the next 15 minutes, so I'm going to be more like just showing you what's possible in the universe of computer science. So one of the problems with what we've been doing in this course, I think it's good to learn how to run the software on the command line step by step. But eventually, you want to scale up. You have 100 RNA-seq files and you want to run them all automatically. So I'm going to show you 2 methods for doing that. For this, you will want to go ahead and get a computational node if you want to try it yourself. If you don't want to try it yourself, just hang out, listen and learn, and that'll be good too.
Will you upload, let's say, sample scripts?
Yes. So if you refresh the homepage, they should be there. They're that lab 6. That has a few samples. If you're looking for more samples down the road, just email me. Miriam and I have been working on moving our RNA-seq pipeline over to Beacon slowly but surely. So we'll probably have a lot more sample scripts in the near future. And if you're on Newton and you just want to be on Newton for the next little while, we've got a million scripts for Newton, so we can definitely help you with that. Okay, so the first thing we're going to do is I'm going to go get some extra files to work with. So I'm going to cd to Lester Medusa, and my username. We have our RNA-seq directory and raw data. Now, I have already added a whole bunch of extra files. You guys don't have these yet. But if you do this copy command, that should give you like 6 extra files. And let me know if that doesn't work. Okay, good. Because I wrote this, I was like writing this up until 6:30 when I left yesterday. And so there's really no telling how all this is gonna work. But you know, that's kind of the reality of working in the command line. You're debugging things as you go all the time. So we'll just chalk it up to being good practical experience if things fail. So if I go back into the analysis folder, I'm going to delete everything I did yesterday so you guys can't cheat. And I guess you wouldn't be able to see it, but I can't cheat. I will have to actually type everything in. Yes. Okay, so I am going to make a directory called O1 FastQC loop and cd into it. And we are going to write a script. And since we only have 13 minutes left, we're going to skip that part about building the script slowly and just build the entire script in one go. So I'm going to open a file called, with nano, called fastqcloop.sh. And into that, I'm going to paste our script. So there's a concept in writing code, and this is not like Python or R, this is just a bash script. So we are writing in the language of the terminal, of the shell. But we can write a for loop. And a for loop kind of means we're going to take this collection of items, whether it's a list of numbers or a list of files or a list of things, and we're going to iterate over it. And for every item in the list, we're going to tell it to do a thing. So for what this is saying is we want to get an ls. We're doing an ls of all of the files in that directory that end in *fastq. We're going to store, one by one, we're going to store that file name into a variable called f. And then inside of the loop, we're going to run fastqc and access the name of the file using that $f variable. So the key thing here is we're iterating over a list of files. For each time we get to a new file, we're going to store it in a variable called f. And then inside of the loop, between the do and the done statements, we can use that $f to reference whatever current iteration we're on. So iterate over all the files. For each one by one, run this fastqc command. So I am going to write this out with Control O. I like that name, and I'm going to Control X. The next thing you have to do is make this executable. So the computer currently thinks it's just a text file. It doesn't know like that you want to run it like it's a piece of code. So to tell it you want to be able to do that, you're going to do U+X. And ch mode means change mode. So change the mode of this file, give me permission to execute it. If you Google Linux permissions, you can go down a bit of a rabbit hole on that, but this will work for now. And then we're going to run it. So this just means within this directory, there's a file that I want to run. I'm just going to put it on the command line like that. And it should start analysis of early flower rep 10. Unfortunately, I'm going to have to kill this because I have like 50 files in there. I was trying to bet that these would run on a whole pile of different sequences, but we'll let it go for a file or two and then kill it and see what happens. So it's gone through the first file, and now it has started on the next file. So this is great, right? Like if I have 100 files, I can now just walk away. If your computer goes to sleep, it can kill your terminal, so that could be bad, but you could be doing other things in the background, and this will just keep running, which is the first way that it's really convenient to get a lot of files processed. We'll let it start on a third one. Okay, so it's on 10, it's on 11, and now it's started on 12. To kill this, like let's say you wrote a script and it was not doing what you expected it to do, you can do Control-C, and you might do Control-C a few times to break that loop. If we do an ls, we can see our files are starting to appear one by one by one. So had we let this just go on, it would have done all 40 or 50 files or however many I have in that raw analysis directory. Okay, so does that make sense? Does the looping make sense? Okay, so like I said, that is a great way to do it if you want to do them one by one, and you're going to keep your terminal open. But the real power, which it's so sad that we're going to talk about this in the last 9 minutes of class, the real power of a high-performance computational system is the scheduler. So you can actually tell the scheduler, I have written this lovely script. I want you to go off and execute it on a computer or on many computers. And I'll come back later and you tell me how it goes. So you can just walk away, you can request a huge pile of resources, many, many nodes, and this is how people who need to process really big data and scale up, this is the real way that you do it. So yeah, I'm gonna skip the star example because we don't have time. If you guys wanna try it on your own, if you have any problems, just let me know, I'm happy to help you out. And I'm gonna bump down to the part of this lesson that says using the Q subsystem. So the important thing to realize about Beacon is that each individual node has 16 cores and 256 gigs of RAM. So when you schedule a job, you are going to get a whole computer. So you get, like, this is a really beefy computer. This is 4 to 8 times as powerful as your laptop, at least in CPUs. So that's, you can run a certain number of jobs on that. Each core can run 2 threads. So I would say on a single node, you could run up to 32 jobs as long as those 32 jobs are only using 1 CPU or 1 thread. And you can kind of figure that out from the documentation. If you're going to run something like Trinity, like an assembly, you can specify, I want to use all 16 cores and I want to use all 256 gigs of RAM. For something like FastQC, for star, we saw that there's a little parameter you can set to use either 1 CPU or 1 thread, or you can set it to use 2, but you have to do the math, right? So you have to figure out how many jobs can I fit on a node. 16 to be safe, up to 32 if it's, you know, you're definitely only running on a single, it only has 1 computational process at a time. So let's back up. I'm gonna make another directory, FastQC, and I think I called it QSub, and I'm gonna CD into that. So again, I just am super organized and I like to keep, at least in this lesson, I'm super organized and I like to keep everything in its own little folder. And let's start writing our scripts. Or actually, I'm just gonna paste it in and we're gonna just walk through what each part of it is. FastQC.QSH. And I like to put the Q to just remind myself that this is something that I'm submitting to the queue, like QUEUE, the submitting to the scheduler. Like I have written the script specifically to be given to the torque scheduler for Beacon. So here's my little script. You'll notice we've gotten significantly longer, but still pretty short, pretty approachable. Up at the top, these are just directions, and we're telling Beacon how we want our job to be run. We want it to use a bash terminal, which is what we have been using in this class. That will almost never change. We want the output and the error files to be generated in this directory. Is that what the J means? We want to run this on a single node. So if you wanted multiple nodes, you could request multiple nodes, although we're still working out how to divvy up the jobs between the nodes. But right now I'm going to recommend, if you use this as a template, stick with 1 node. And these FastQC jobs run super fast. So I just gave it a wall time of 7 minutes. Like, this is going to be a really fast job. We have to change directories to our working directory. So once we submit this and it goes off to run on another computer, it doesn't know where you were when you submitted it unless you tell it. Like, go ahead and change them to the same directory that I was in, that the script lives in. And then we have a very, very similar for loop that we had earlier for the files in this directory that end in FastQ. I use this little command called basename, which strips off the path. I echo the file name, and the host name and put that into an output file, which we'll just use to see where it ran. And then I have my fastqc command. So these are, it's a bit of an art form to write these. There's a lot of training sessions that you can go to with Nix and Jix to write these and to learn more about it. There's more classes you can take. But this is kind of a basic example. This will launch like 40 jobs. So we're gonna launch it and see what happens. I probably before this lesson should have cleaned out some of those. Well, I guess I could delete a few. No, we'll launch it, it'll be fine. These fastqcs run so fast, it'll be fine. X, and then q sub fastqc.qsh. So this q sub command basically just means stick this out on the beacon. I want it to run, I want to submit it to the scheduler, that's what the sub is for, and let it just go run somewhere else. So when I do that, oh, I forgot to specify our project. So at the top, you can use the PBS-A U10 and our course number. I scheduled this under the default opportunistic cycle, so it might take a little while to run. But that's something you can specify at the top. And then to see how your job is doing, you go to qstat. And this tells you all these other people are running things on Beacon at the same time. You guys all have logged in to a terminal, so I can see that. My job is down here, fastqc.qsh. I can see that it is starting to run. So R means running, Q means it's queued and waiting. So we can keep typing qstat. It's still running. But I'm starting, if I do an ls now, I'm starting to get tons of output. So it's running. I can tell it's running because it's generating output, and I'm starting to see HTML and zip files, so my fastqc is running. So you can kind of just monitor this. I think you can also set it up to send you an email when it's complete, which is really nice. Yes, M. Reynolds has been using the computer for a while. That's what that means, right? It's been running 1,479 hours. Or it's used that much computational time split across different nodes. So if you had, it's only been running for 20 hours, but it had been running, or it had only been running for 14 hours, but it had been running on 100 nodes.
He's running a particularly long job. They do climate research in air quality for the state of Tennessee. So they've been running a really long job to figure out what air quality in Tennessee. If you're in Knoxville, it should be longer than that because everybody's got to.
Right, they really need to really do that research. That's good. That's what we want our computers used for. Fix the allergies.
Yeah, we're all like, yeah, need a little more time. Coughing and taking care of that.
Right.
Yes, take what time you need.
They have a really powerful argument. But this is great. Like we're sharing this computational systems with people who are doing medical research, engineering, physics, like computer science, all kinds of stuff. So that's really the beauty of having an HPC. Let's see. Looks like it's still running. But again, now I'm really seeing all my HTML and .zip files are just piling up in this folder. So I didn't have to sit there and type that command 40 or 50 times. I created my script, I submitted it, and it's off running. And if we cat, I'm gonna output all of the .output files. And if you'll remember, when I wrote the script, I put in this kind of like thing we haven't seen before, but I wanted to echo the file name and echo the host name, so we could see like what computer did it run on. That's just fun to know. So I'm on Beacon 3, but all of my jobs got run on Beacon 2. So they can get run on another computer than the computer you're on. So that's kind of the nice thing about a scheduler. Yep, still running. So I think it'll get killed at some point because I only asked for 7 minutes. But so that's like the super high level overview of how the batch systems works and how the queuing system works. You could also request a whole node to do one job. You don't have to have a for loop in that script. You say, I'm doing an assembly. I want to use all 16 cores and 256 gigs for this one thing to just do my assembly. You write your little submission script and off you go. The limit right now is 24 hours. So if your job runs more than 24 hours, it'll get killed. It sounds like you might in future be able to email the help desk and be like, please.
If you're doing something to help my allergies, yeah, you people are something.
So you should lie and say it's going to help his allergies. No, I'm just kidding. You should make your research sound super compelling. Yeah. So that's my advice. If you guys need help setting up submission scripts in the future, my lab, we were sitting in the lab working on this for yesterday afternoon. We're going to be doing a lot of this for RNA-seq, so just come talk to us. We can probably help you out. Any questions? That was like the fastest I've ever tried to teach Q-sub and scripting ever. All right, if you're an enrolled student, don't forget to send me your 2-page report on your paper by next Friday. End of day, it's fine. I haven't gone through and graded all the emails to make sure you sent me materials from each lesson. I will do that. But if you happen to did an email earlier, feel free to email them to me. I'm not going to count that against you. And thank you.